body texture B and color is represented by small r, what will be the genotype of the female parent? And if recombination is considered, then what are the genotype of all the progenies of the F1 generation? So whenever you get questions like this, first you have to identify which is your dominant gene and which is your recessive. Okay, so in your question, it's already given here that smooth body and gray color are the dominant traits, and bumpy body and white color are the other recessive ones. So bumpy body is recessive, white is also recessive. Okay, so after identifying which are the dominant ones and which are the recessive ones, you first make out which is the male, then write down the genotype of the male and write down the genotype of the female. So the male here is written here that the male beetle, the male beetle has recessive genes for body texture and color. So the male beetles is going to be this one. So since the male is homozygous recessive, so its genotype is going to be this one, like small b and small r and small r. Okay, so now for the female, because the female is heterozygous. So since the female is heterozygous, like the genotype is going to be capital B, capital R, small r. So any form which you're writing like this, like one dominant gene, one recessive gene, this is called as the heterozygous form. Okay. So the first question has been answered. So what is the genotype of the female parent? So if recombination is considered, then what will be the genotype of all the progenies of F1 generation? So now you have the male genotype, you have the female genotype. Now you have to perform crossing between these two. So from the female, you are going to get two kinds of alleles. Like one is capital P, capital R, and one is going to be, sorry, you are going to get four kinds of alleles, sorry. So one is capital B, capital R. One is going to be capital B, small b, capital R, and small b, small r. Okay, so from the female, you are getting four kinds of alleles. Now from the male, since the male is homozygous recessive, like you are getting only one kind of alleles, which is small r, small b, and small r. So now for constructing this, the genotype of all the progenies, like from the male, you are getting only the small b, small r, and from the female, you are getting these four kinds of alleles. So now just make the cross between these two. So the first one, which is capital B, capital R, small b, small r, you are getting this capital B, capital R, small r, which is the same as the female. Then the second one, which you are crossing with capital B, small r, and small b, small r, you are getting capital B, small r, small r. And similarly, you do the rest. And accordingly, like you just figure out what is going to be the phenotype. So the capital B, it means that, so capital B means it's the smooth body. And the capital R means that is a gray color. Small b means is bumpy. And small r means white. So accordingly, you can put the phenotypes next to it. Okay. So another problem on linkage and crossing over is that you have two genotypes which are given to you, capital A, capital B, capital B, and small a, small b, small b. So one is homozygous dominant, another one is homozygous recessive. Okay, these two were crossed, and then the F1 progeny is again crossed with the homozygous recessive one. So the question is, what percentage of the second cross progeny will be homozygous recessive if the two genes are completely linked? So in the previous example, the genes were not linked. That's why you could get multiple different kinds of alleles. But since, okay, this one is linked, you are not going to get multiple kinds of alleles. So I'm going to show you what's happening. So this one, like capital A, capital B, is going to give you allele is capital A, capital B, and small b is going to give you the allele small a, small b. So when you cross these two, like the only outcome you're getting is capital A, small a, capital B, small b. Okay, now you are crossing this one, the F1 generation, capital A, capital A, small a, capital B, small b with the homozygous recessive. So now since the genes are completely linked, like in the previous, if the genes were not linked, then from this combination, you would get four kinds of alleles. Like in the previous example, like you had the heterozygous form, you got four kinds of alleles. But here, since the genes are linked, you will get only two kinds of alleles. which is capital A, capital B, small a, and small b. And from this one, again, you will get only small a and small b. So when you're crossing
Why not?
both,
Why not capital A, small b? How will
yeah,
you know
because
capital A, capital B?
so here you are not going to get these combinations. You are not going to get these two combinations because the genes are linked here. The genes are completely linked. If the genes were not linked, then you would get all four of these. Okay. But since the two genes are linked, it means that A and B, they capital A, capital B, they always go hand in hand. Small a and small b, they always go hand in hand. So this is what linked means.
Could you please
So if
explain in a chromosomal manner what was happening?
okay, I'm running out of space here. So this is a chromosome. Suppose you have this is these are the chromosomes. Okay, so when you're having recombination, during meiosis, it means that the genes they like it can happen like this, like these can recombinate over, these can recombinate over, or like this, like this, like this, like this. It can happen like in multiple different ways. But if the genes are linked, in that case, what is going to happen is that again you have the chromosome. Like you have one gene here, one gene here, one gene here, one gene here. So it means that these two genes during meiosis, they always go together. Like this crossing over, like with this gene, this thing which is happening here, the crossing over, it doesn't take place. That is what linkage means. So in this example, like again, the chromosome, suppose this is capital A, this is capital B, this is small a, this is small b. Since these genes are linked, they always go together. Like this recombination, it does not take place. Like do you know what is recombination? Has that been taught
Means
to you in the
crossing over, right?
Yes, so like crossing over or recombination, that is a feature of the meiosis cell division. So in meiosis cell division, like it's that is the differentiating feature between meiosis and mitosis, that there is something called crossing over or recombination, like there is exchange of genetic material between the chromosomes. Okay, so this exchange which is happening between the two chromosomes, this is what we call as recombination or crossing over. And when the two genes are linked, that means the crossing over, it does not take place. Means these genes, they are always transferred as it is. That is what it means. Like means one gene on one chromosome, one gene on the other chromosome, they are not like recombination does not take place. They are always transferred together. Like, did I make myself clear? Like, if you have any doubts, just ask me.
So if the genes are completely linked, then the dominant will always go to the dominant and recessive will always go to the recessive. Capital A, capital B, small a and
Yes.
small b, right?
Yes.
Okay.
So when they are completely linked, the homozygous form, like capital A, capital B, small b, like the dominants always go together, recessives always go together. You are right about that. Okay. So now from this crossing, like this cross, you are getting three kinds of alleles. Okay. So just to, construct the chart. So like when doing these kinds of questions, it's a good idea to always construct the chart to avoid making any mistakes. Okay. So you have capital A, capital A, small a, capital B, small b, small a, small a, small b, capital A, small a, capital B, capital small b, small a, small a, small b. So we can see in 50% of the cases, you have the heterozygous form, which is capital A, small a, capital B, small b. And in 50% of the cases, you have small a, and small b, small b. Okay, so is this question clear with everybody? If you have any
Yes.
doubts, Okay, so like we can move forward. Okay. So a cross is made between a pure wild-type male and a brown eye curled winged female of a hypothetical fly. Then a cross test is done with the F1 progeny. This cross gives you the F2 progenies as follow. So this is just a numerical question, like you don't have to construct any chart here, but you have to know what is the recombinance. So the wild type is this one. So your wild type is this. This is your wild type, and this is the female. Okay. So the wild type and the female, both of these, they are the homozygous forms. Okay. Okay, so this is the male and this is your female. So both the male and the female, they have the homozygous characteristics. So obviously whatever progeny they are going to form, they are the recombinants. So out of the total here, like the total is how much? The total is 300. So total number of progenies are 300. So out of them, your recombinants are 12 plus 8 is 20. So the question is, what is the recombination frequency? So recombinant frequency is your number of recombinants by total progeny into 100, which is, okay, so which is that? 20 by 300. So 20 by 300 into 100. This is your recombination frequency. Okay, so is everybody clear on this sum? Okay, so moving forward. Okay, so this is your pedigree chart. So like just, can someone just clarify like whether you have learned about the basics of pedigree? Like what do these symbols mean and everything? Do you know that?
No.
Okay, so this is brand new to you. You are seeing this for the first time.
Yeah.
Okay. So in a pedigree, like a circle, this means female. Square means a male. When anything is colored, like completely colored like this means the person is affected. So in this case, the circle is completely colored. This means the affected female. If it's a square and it's completely colored, then means this is, yeah, Diksha, can you just like, when the clock hits 7:45, just stop me, okay?
Yeah, sure.
So this is affected male. Okay, and when it's half colored, like this is a carrier, male. And when it's half colored, this is carrier, female. Okay. So until now, all the examples you were seeing, they were not clinics based. Okay, those were just, experiments or genetics carried out on flies or some other animals. But the pedigrees, they are entirely clinical based. Okay, so here, like you don't have to remember, like you have to remember what is homozygous and heterozygous, but these are purely in terms of clinical basis. Okay, so sometimes the disease, like some diseases, they can be homozygous, they can be heterozygous, it doesn't matter. But the main things which you have, the main terms which you have to remember here are the terms dominant and recessive. So sometimes you have genes for a disease. Like if the gene is a dominant gene, that means the person, like if that person has one copy of the gene itself, that means the person is going to be affected. Okay, so out of the two genes, if one gene is a dominant gene and that gene is responsible for the disease, that means the person is going to be the affected person. And if the disease, sorry, if the gene for the disease is a recessive gene, it means that the person needs to have both copies of this gene to be affected. Otherwise, if the person has only one copy of this gene, it means that the person is going to be a carrier. Okay, so just keep these things in mind and we'll proceed with the questions. Okay. So what's like? Okay. So taking the example number 8. Okay, so here in the question is already given here that, okay, another thing which I want to say is that So in case of the pedigree analysis, like there are two kinds of diseases you have to know. Like one is X-linked and one is autosomal. Okay, X-linked means these are related to the X and Y chromosomes. Okay, like these are the sex chromosomes. So these are gender-based. These diseases are always gender-based. And autosomal means these are the general body chromosomes. Okay, not the gametes. So these are have no gender. Like they can affect persons of any gender equally. Okay. So here in the question is already given here that the disease which you're dealing with is X-linked. And you just have to identify whether the pedigree is of the dominant or of the recessive. Okay, so taking example number A, here in the first generation, you can see that the female is an affected female. Okay, so and in the second generation, again, you are saying that the female is affected. In the third generation, again, the female is affected. So as a general rule of thumb, if you're saying that a person in every generation is being affected, it means that automatically it is a dominant gene. And here you are seeing that all the females specifically are affected, like none of the males are affected. So automatically this becomes X-linked dominant.
Shatrupa, I think we can stop here. It's 7:44, so people need to go.
Okay, so like you have your quiz now from 7:45 to 8. So after finishing your quiz, you can join back for the rest of the tutorial.
Hello.
Yes.
Now we have to like launch the safe exam browser or something. Now we
So,
have to launch the safe exam browser.
launch. If it is instructing you to launch, then you have to launch it. Have you logged into the model? Hello?
Quiz was on the Moodle, right?
Okay. Guys, if you're back, can we start again?
Like there are only 9 people.
Yeah.
The total was 21. No?
Yes.
Okay, so let's wait till 8:05.
Sure. Okay.
I have a doubt. In this quiz, there was a question given that is seen exclusively in case of boys in a family and also seen in each generation. What is the possible mode of inheritance? Why this can be X-linked inheritance, right? But they have given Y-linked inheritance. I mean,
Can you
X is
repeat the
the question is that the disease X is seen exclusively in case of boys of in a family and also seen in each generation. What is the possible mode of inheritance for this trait?
it's X-linked?
But they have given Y-linked because I thought X-linked and I kept, but they have written it as Y-linked.
Okay. Okay, so
Uh,
it's both, I guess, because in X-linked also, like, it's like not exclusively, or it wasn't mentioned that it's exclusively seen in males.
yeah, exclusively in case of boys and also seen in each generation.
Okay, then it's violent only, because if it's X-linked, then it's mostly seen in boys, but it can affect the females also. So because that exclusively word is there, that's why it has become violent.
Okay. And if we do like if we cross between a black rabbit and a white fur rabbit, we'll get
Yeah.
spotted or gray one.
Like what's the dominant and what's the recessive in this?
No, both are dominant. I mean, both are heterozygous. Co-dominant. Both are dominant.
In the question, it was given that co-dominance,
Yeah.
so it will be spot.
And I guess it should be spotted only.
Yes.
Okay.
So well, the answer is spotted, right?
Yeah, it is given
Yes,
spotted, but the doubt is like, why can't it be gray? Like in a flower, they gave it is as a color mixture sort of thing, right?
Because yeah, because if it would be gray, then it would be incomplete dominant. Okay, so it is co-dominant. That's why both the traits are expressing themselves. That's why it is spotted. Both black and white are being expressed.
Okay, okay.
Anything else?
Yeah, so let's start. It's already 8:07.
Yes.
Okay. So I'm just starting to share my screen. Okay, so we were on the slide. Okay, so in this one, in example number A, since all the females in all the generations are being affected, so this one is X-linked dominant. So by default, when you're looking at any pedigree and you're seeing that people in every generation is being affected, then by default it automatically means that it's going to be a dominant disease. It's not recessive. Okay, and when you're seeing exclusively only one gender is being affected, like here you can see that the other generation, other people, they are not carriers or anything. So that's why, this one is X-linked dominant. Like in case of the females, suppose if only the males were colored like this, then that would have been Y-linked dominant. So I guess like in your question which you got, like if the pedigree was given like that, only the males are colored, none of the females are carriers. Like if it was X-linked dominant, then sorry, if it was X-linked recessive, then the males would have been colored, and then a couple of females, they would have been carriers, like half colored like this. But since if there is no half coloring like this, then that means that that would have been Y-linked dominant. If some of the females were carriers, then that would have been X-linked recessive. Okay. So in this one, in example number B, and another thing also, like, in the some of the pedigrees, like they don't show the carrier one. Like in this one, like this example number B, it's of X-linked recessive. Okay, but they have not shown the carriers. So I'm just going to show it for you. So here in the first generation, you are seeing that the male is not affected, female is not affected. But in the second generation, the male has become affected. So what this means is that the female is a carrier. The female is a carrier, so that's why the X, suppose the female genotype is this. Suppose this is hemophilia. Just suppose this is a gene for, this is the pedigree of hemophilia. So the female is XHY and, sorry, XXX. So this is the hemophilia gene. This is the recessive one. So the female is a carrier for this. The male is XY. So this affected male, he is XHY. Okay, so why is this male suffering? Because he has only one copy of the X gene. Okay, so the Y chromosome is not having any effect on this X chromosome. So since this male, he has the hemophilia gene on the X chromosome, so this male, he is going to suffer from hemophilia. Okay, and this hemophilia gene, it was passed on to him from his mother. Now, since the mother is having one normal copy of the X chromosome, so that's why the recessive hemophilia gene is not being expressed. That's why we are saying that the female here is a carrier. Okay, now here in the first generation, the male is affected. He is getting married to a female. And then in his progeny, one of the females here is affected. Okay, so since one of the females is affected, it means that this person's mother, the mother is also another carrier here. Okay, so now if you work out the genotypes, you will see that in this case, this affected female has both copies of this, suppose this hemophilia gene. So both recessive copies, this female is suffering from hemophilia here. And all the females, like when you cross this, all the females, they are going to be carriers. Like all the females are either going to be carriers or they are going to be affected, which means that this female is also another carrier. And when you cross this, you will see that in 50% of the cases, the male is going to be affected. And in 50% of the cases, the male is not going to be affected. Okay, sorry, this is I should not have made the here. Okay, so here if you cross this, you will find that in 50% of the cases, the male is going to be affected. And in 50% of the cases, he's not going to be affected. So here, their son, he has not been affected. Okay. Now, in the fourth generation, this carrier female, she has married an affected male. So the affected male is, suppose this. This is the female. And in their children, they have one affected daughter. Okay, so the daughter is this one. Okay, so just by luck, none of their sons, they don't have the disease, but in 50% of the cases, they could have had the disease. In 50% of the cases, they may not have had the disease. Okay. So, but again, like all of their daughters, they will either be carriers or they will have the disease. So, which means that this daughter again is a carrier. Okay. So, did everybody get this?
Could you please explain A one more time?
This one, A.
A.
This one, example number A, the X-linked dominant one.
Yeah.
Okay. So X-linked dominant, okay, suppose it's so suppose this is like the X-linked dominant disease, one is called is Duchenne muscular dystrophy. Suppose this woman is suffering from that. So I'm representing it by XM. So this XM, this gene is the dominant gene. And is responsible for the disease which I'm calling as muscular dystrophy or Duchenne muscular dystrophy. Okay. So she has one copy of this gene. So since it's a dominant gene, that means that even if she has one copy of it, she is going to be affected. Okay. And this man, he is not affected. Now, when you cross this, just mentally cross it. So in 50% of the cases, you will get a normal female. And in 50% of the cases, you will get an affected female. You had an affected female. And in 50% of the cases, you will get an affected man. And in 50% of the cases, you will get a normal man. Okay. So this female, she is affected. Again, she is this one. Same X. This man, he is normal. This woman is normal. Okay. So this a woman, she's marrying a normal man.
Okay, I got it.
So again, you can just mentally do it. Okay. So this one is again similar to the first generation. Okay. This cross. So is everybody clear with this one? Okay. So moving ahead. So this is just the same thing which I just showed. Like they have showed it. Okay, so another one is what type of inheritance is of the affected population. Okay, so in this pedigree, they have shown the carriers as well. So in the first generation, okay, so just by looking at this, okay, can anyone guess whether it's autosomal or sex linked?
I think X-linked.
Yeah, I think so too. Yeah, it's X-linked. Because you can see that the males, they are affected. This male is affected, and all the females, they are carriers. So this is typical of the X-linked recessive. Yeah. So it just is written here what I just explained, that it affects the males because they have only one copy of the X chromosome. The females, because they have the two copies, they are mostly the carriers. This is what is shown, in case of the genotype. Okay, so this is an autosomal one. Okay, so they have shown it for phenylketonuria. So this is an autosomal recessive disorder. So for an autosomal disease, you can see that it affects both the genders almost equally. So men are also carriers, women are also carriers. Men are getting affected, women are also getting affected. So this means that it is an autosomal thing. We have not decided yet whether this is recessive or dominant. But in case of an sex-linked one, like whether X-linked or Y-linked, like in those cases, like either the male is going to be a carrier or a female is going to be a carrier. Like never, both the genders can never be a carrier. Okay. But here you can see the first generation itself that the female and the male both are carriers. So automatically this means that this is autosomal. Okay. Now this concept of a carrier, this comes only when the thing is recessive. Okay, because like if the gene is dominant, that means if the person has one copy of the gene, that means the person is going to be affected. So there is like the person can't carry the gene. Okay, so if the gene is recessive, it means that if the person has one normal gene, one recessive gene, that means the person can transmit that recessive gene to the next generation. So that person acts as a carrier. So whenever you're seeing the carriers like this, automatically it means it's recessive. So just by looking at the first generation, you can just make it out that this is autosomal recessive. Okay, so now just explaining the pedigree. So the the carrier female is meeting with the carrier male. So in the first generation, they have got an affected son, affected daughter, carrier male. Okay, so then they are again meeting and you are getting something like this. So what is the probability that 12 is a carrier? Okay. Okay, so just like I'm just assigning some genes to it. So suppose this is the normal form. The small a is the recessive gene. Okay, so small cap, this is going to be the carrier. This is going to be the affected. Okay, so this person is this. Person is this. Okay, so both of them, so by just crossing these two, you can see that both of them are going to be carriers. So what is probability that 12 is a carrier? So this is going to be 100%. Okay, then what is the probability that 16 is genotypically normal? Okay, so carrier is mating with affected. So this chances of this can be A or A. So these are the two possibilities. Like from the mating of this, these are the two outcomes. So what is the probability that 16 is genotypically normal? Okay, so in 50% of the cases, this person is going to be normal. Okay, so this is 50% cases. And in 50% of the cases, the person is going to be affected. Oh, sorry. So they are genotypically normal. So sorry. So here's the so I just want to clarify one thing. So there is one concept of genotype, there is one concept of phenotype. So if the question was phenotypically normal, then this would have been 50% only. Because carrier and a normal like this, because a carrier, somebody who is a carrier and somebody who is normal, their phenotype is the same. But since they are treated is genotypically normal, that means a genotype, they have only considered this one, capital A, capital A. Okay, so in these outcomes, like there is no capital A, capital A. Okay, so that's why in 0% of the cases, the person, there is never going to be a genotypically normal outcome from this. But if the question was a phenotypically normal, then it would have been 50%. Okay, sorry, I made a small mistake here. So since there is no capital A outcome of this, so that's why the answer of this is going to be 0%. Okay, so I hope there is no confusion here. Like, are there any doubts in this slide?
Actually, a small doubt that
Yeah.
once we have identified the disease is autosomal disease, and so
Yeah.
why there is a need to classify the subjects as male and female?
Like you don't have to classify as male and female, but it's just good practice to just identify the things which you're going along. Like I have been doing this for many years. That's why, like by habit, it has become like this. Like I'm identifying, just looking at this, I'm saying this is male, this is female. But if you're not very confident, not fluent with it, then there is no need to, just put mental effort into identifying a male and female separately. Just if it's autosomal, you can just do it in a carrier or recessive or normal kind of way. But with when you're doing this over and over for many years, then it just becomes fluent like this. Okay, so there is no compulsion to identify male and females in this case. Okay.
Yeah, okay, got it.
All right, so moving ahead. So this is just what they the thing which I showed. Okay, in this pedigree chart, what is the type of inheritance observed, provided generation 1 genotypes are unknown? Okay, so the generation 1 genotype is not known. So by just looking at the remaining genotypes, you can see that in every generation, people are getting affected. So by default, this is dominant. Also, you're seeing that every gender is being affected, males and females. So it's autosomal. So the answer, it should be autosomal dominant. Yeah, autosomal dominant. So it's the same thing which written here, as the mutation is observed in all generations in both the sexes. Okay. The same thing, like they just showed out the genotypes here. So did everybody understand this one, why I'm classified as dominant and autosomal? Because you're having affected people in all the generations, is affecting both male and female equally. So that's why I just said, it's good practice to, to identify males and females, because just by looking at this, you can quickly identify which is a female here and which is a male here. Okay. So since it's affecting all the genders, that's why it is autosomal. Okay, we are done. So any other questions? Any questions about pedigree? No doubts at all?
Actually, I had a trivial doubt, but it was in the co-dominant part.
Yeah.
Where the picture was shown of a flower with red and white, both colors are appearing.
What picture did they?
You can go to slide 2, I guess. Yeah, it is slide 2.
Yeah, so in
slide
the
3.
picture, it was shown that the circle, they were radially, red color was in the inner ring and white color was in the outer ring or somewhat, or the other way around. So, like, why was the specific order, like first red color is inside and then white color is outside of it?
Actually, here the expression is shown that both the traits are expressing themselves. The pattern is not important here. The pattern could be anything, like it could be spotted, it could be like the red is on inside and white is outside. Okay.
So yeah, I was just asking how the pattern is decided then.
How the pattern is decided, actually it depends on other genes as well. So how the genes are interacting with each other. So it's not just that these genes will decide the pattern only.
Okay, fine. Thank you.
Okay, so if there are no doubts, I guess we can end. Okay, Diksha, we can end the session then.
Yeah, sure, we can end. So you guys can leave.
Just like I just want to ask something, like should I share this annotated PDF, like this annotated PPT with them or the original one?
We can share the original one. Is everybody okay with that?
Like if you want the annotated one, then I'll just save this as like the annotated one. Otherwise, I'll delete it.
Can you share the both PPTs as well?
Okay.
Okay, sure.
Like label one as annotated one so we can identify which one is which.
Okay.
Yeah.
Thank you.